Good morning, everyone. I hope you're having a wonderful weekend. The title of my talk is Designing Potential Extensions from the Global Substance Registration System to the ChEBI Ontology to Identify Natural Product-Drug Interactions. I'd like to thank the Bio-Ontologies community for giving me the opportunity to present this work today. Most of us use one or more natural products in daily life. These can include dietary supplements, food items, or herbs for pain relief. The FDA groups these together under complementary health approaches, and the internet is full of information about the benefits of various natural products like green tea, cinnamon, grapefruit juice, and many more. There are other natural products under this umbrella that are taken as alternative medicines. For instance, kratom is an herb used for pain relief, and cannabis that has been legalized in many states in the United States now. With the increasing use of these products, interest in research related to natural products has also increased. Looking into the availability of natural products in current biomedical ontologies, we come across three main resources: the Food Ontology, the Drug Ontology, and the National Cancer Institute Thesaurus. Taking the example of green tea, a popular natural substance, information available about the product varies depending on the source that we're looking at. It generally includes hierarchical information about the natural product, its synonyms, semantic types, and some external references. In certain cases, such as in the Food Ontology, there may be annotations related to the quality, formation, and derivation of the product, and drug capsule information that can be found in the Drug Ontology. When comparing this knowledge to information generally available about common drugs, we can see a major difference. For instance, simvastatin is a drug used to treat high cholesterol and has extensive information available in the ChEBI ontology related to its chemical properties, biological roles, applications, and much more, in addition to the information we talked about before with natural products. A key difference is the availability of pharmacokinetic information about the drugs, including its metabolism, targets, and the participating enzymes. Pharmacokinetic information is useful and valuable for researchers when looking at pharmacokinetic drug interactions. By pharmacokinetic drug interactions, I mean interactions that involve a drug A and drug metabolizing enzymes and transporters that lead to increase or decrease of the concentration of a substance in the body. In case of natural product-drug interactions, this drug A is instead a natural product that affects the enzyme or transport protein in the body and thus interacts with another drug B. For instance, in case of grapefruit, it was found that one whole grapefruit or a small glass could cause drug toxicity as the constituent of grapefruit inhibits the cytochrome P450-3A4 enzyme, and so any medication such as simvastatin that is metabolized by this enzyme and has low bioavailability would potentially lead to adverse events. Bringing this back to bio-ontologies used to represent such knowledge, the limited availability of pharmacokinetic information about natural products can prevent rigorous research about natural product-drug interactions using current ontology resources. For instance, our particular use case involves identifying plausible mechanisms for natural product-drug interactions such as these, using a knowledge graph created from existing ontologies and biomedical literature. And thus our goals in this study were to integrate biomedical data about natural products in the open biological and biomedical ontology foundry ontologies, design potential logical extensions to include natural products, natural product constituents, and its related pharmacokinetic information in biomedical ontologies, and eventually facilitate the discovery of potential natural product-drug interactions. We followed a simple workflow in the project, including extracting data about natural products, generating semantic representations, creating logical statements, and finally working with the enhanced ontology. We chose to design potential extensions to the ChEBI ontology instead of creating a separate ontology of natural products for multiple reasons. First, as we saw, the ChEBI ontology is already a major source of information related to chemical entities such as drugs and some natural product constituents. It is also an established ontology within the OBO foundry and is extensively used in research. We further wanted to take advantage of the interoperability between the OBO ontologies, as you'll see in the upcoming representations. And thus, with ChEBI as our base ontology for the extensions, we set out to identify the best data source for information about natural products and their constituents. To design the potential extensions, our information requirements included standard identifiers for the natural products, access to domain-relevant information such as the genus, species, constituents, and their chemical characteristics, a comprehensive coverage of natural products available on the market and in use around the world, non-ambiguous names for the natural products, and the availability of pharmacokinetic information. We looked at a number of potential data sources for these information needs, including the Global Substance Registration System, the Dietary Supplement Label Database maintained by the FDA, the Licensed Natural Health Products Database based in Canada, the Unified Medical Language System, and some other sources traditionally used for information about drugs. We used the Global Substance Registration System to extract information about natural products due to the wealth of information available about natural products, their constituents, and their properties. The G-SRS is led by the GNAS project, and it harmonizes chemical identification for drug development and safety research using an ISO standard. It is the primary substance identification system used by the FDA and contains over 25,000 structurally diverse substances or natural products in the system. In addition to natural product constituents and metabolites, it also contains external database references to the Dietary Supplement Label Database and National Cancer Institute Thesaurus, and the information generally is verified by experts. Prior work has also found that the G-SRS contains the largest number of synonyms of known natural products along with the Dietary Supplement Label Database. While the Global Substance Registration System does contain extensive information about natural products, we also extracted data from the Center of Excellence for Natural Product Drug Interaction Research, or the NAPTI Center, to include current pharmacokinetic information from the literature and clinical studies. This center was set up by the National Center for Complementary and Integrative Health at the NIH to assess clinical relevance of pharmacokinetic natural product drug interactions and address existing gaps in scientific literature about the same. It also contains a publicly accessible data repository with scientific results, raw data, and recommended approaches related to pharmacokinetic and PDIs. Armed with all this information, we then generated semantic representations to include in our enhanced ontology. This was done by adding novel classes for each natural product and its parent substance, and then determining the class-class and instance-instance relationships that exist between the entities. We used the relation ontology, ChEBI, and basic formal ontology terms to establish relations between the different entities, and then generated semantic representation patterns based on the available information in our data sources. For instance, based on whether or not we can find classes already in the ChEBI ontology, we had different patterns, such as where the natural product constituent class exists in both the ChEBI ontology and in G-SRS, versus if it only exists in G-SRS. And similarly, if a natural product metabolite class already exists in ChEBI ontology and not. I will now walk through one of the patterns to explain our approach where the natural product constituent class already exists in the ChEBI ontology. So we started with classes for the natural product and the natural product parent. In these representations, rectangles represent classes and ovals represent instances, which are related to each other through the RDF type relationship. The natural product classes are linked to SRS through the database cross-reference relationship, followed by relating the natural product and natural product class to the plant ontology through the plant anatomical entity. An instance of a natural product is related to the natural product parent instance using the part of relationship. For example, a green tea leaf is a part of the green tea product. This was followed by including constituents of the natural product, and in this case, this constituent already exists as a class in the ChEBI ontology. It is also linked to SRS through the database cross-reference relationship and is a subclass of the chemical entity in ChEBI ontology. The constituent is related to the natural product instance through the has component relationship. We then included information about the natural product metabolite in the representation, In this case, the NP metabolite did not already exist as a class in ChEBI, and so we created a novel class that was linked to the metabolite in G-SRS. Each natural product metabolite, whether or not it exists in the ChEBI ontology, will be a subclass of the chemical entity in ChEBI and will have a role of metabolite. It will also be related to the natural product constituent through the has functional parent relationship. We further include information about pharmacokinetic studies about the natural product and its related organism. The colors in this final representation are only to differentiate where the information or relationship is coming from. However, we believe that our extensions to the ChEBI ontology to include information about natural products from the G-SRS and other Bio-Ontologies can cover a wide range of valuable information. As an example, here is one of the representations we have created for green tea with a constituent epigallocatechin and its metabolite that both already existed in the ChEBI ontology, but can now be related to more extensive information in the G-SRS. I'll pause for a moment so you can take it in. Once we've generated the semantic representations, we moved on to the last two steps, which was creating logical statements for the semantic representations, and then finally creating our enhanced ChEBI ontology. To generate the logical statements, we translated the semantic representation patterns to logical statements in the web ontology language, and then serialized the statements using the RDF-lib library in Python to RDF XML triples. We used blank notes to represent existential variables. As an example, the 4 triples on this slide represent the 4 relationships from this subset of the representation for green tea. To create the enhanced ontology, we merged the triples created for the natural products with the ChEBI light ontology, and then applied the Hermit reasoner to verify its logical consistency and infer any novel axioms. Our enhanced ontology currently contains representations for kratom and green tea while we work on generating statements for other natural products, but it has over 156,000 classes and over 1 million axioms. We're also working to generate patterns and extract data about natural products in a more automated manner for comprehensive natural product inclusion in the ontology, as we currently have a list of over 600 natural products that we would like to include. We also want to differentiate between in vitro and clinical pharmacokinetic results while making sure that the sources of this information are also included. We hope to create evidence strategies for the knowledge using artifacts from the DDEO and CPO ontology, and then finally include this enhanced ontology in a large-scale biomedical knowledge graph combined with machine reading for hypothesis generation for natural product-drug interactions. Thank you all so much for listening. I would like to thank my co-authors Richard Boyce, Tiffany Callahan, Matthias Brockhausen, Mary Payne, and Sandra Kane-Gale for their support. I am happy to take any questions now. Thank you.